Hello everyone, today we are here to discuss the previous year NEET questions on locomotion and movement. So this is the part 1 video. So moving on to the first question. Lack of relaxation between successive stimuli in sustained muscle contraction is known as option A spasm, option B fatigue, option C tetanus, option D tonus. So lack of relaxation between successive stimuli. Lack of relaxation is a pathological condition. Pathological condition which is caused by a bacterial infection, which is called tetanus. Which is caused by a bacteria Clostridium tetani. So this is also called lockjaw disease. That is the mandible, that is the lower jaw gets stuck. There's a continued contraction without any kind of relaxation. It's a painful contraction, and so it is called sustained muscle contraction. So the answer is tetanus. And so let's discuss what the other three definitions are. Spasm. Spasm is also a muscle contraction, but it is not sustained. Spasm is when you're gone for a long walk or a long run and suddenly you have a sudden muscle pull. That's when you have a spasm, a sudden contracture of muscle, and it's relieved later. What is fatigue? Fatigue is the tiredness of muscles. That is when you've gone for a long walk, or when you've started doing an exercise, you suddenly feel that you can't do the exercise anymore. You feel weakness of the muscle. So there is depletion of ATP, and so there's accumulation of lactic acid through anaerobic pathway. So this leads to muscle fatigue. What is tonus? Tonus is defined as the minimal activity of muscle tissue in the body even when the body is in rest. That is, so when we think that the muscles are at rest when we are asleep or when we are not having any action, but that is not actually true. When you're asleep or when you're not in any action, actually there is a small amount of we can't feel it, but there is a small amount of tone, that is where there is a contraction, relaxation that happens inside the muscle. So this is called the natural tone or tonus of the muscle. So this is seen in all types of muscles. So the answer is tetanus. Sustained contraction with no relaxation phase is called muscle tetanus, caused by Clostridium tetani. So moving on to the next question. Sliding filament theory can be best explained as option A, actin and myosin filaments shorten and slide past each other. Actin and myosin filaments do not shorten but rather slide past each other. When myofilaments slide past each other, myosin filaments shorten while actin filaments do not shorten. When myofilaments pass each other, actin filaments shorten while myosin filaments do not shorten. So let's look at the explanation. So this is the sliding filament theory by which we have the muscle contraction. So we have actin and myosin filaments, myosin head. So we have this myosin head here, which will split ATP into ADP and a phosphate. So there's a utilization of ATP here. So this head will go and bind there to the actin filament. So then there is this stroke. So there is constant movement of this muscle, and then after this is done, when the ADP is released, again there is the relaxation. So the answer is the actin and myosin filaments, actually they do not shorten anywhere. We don't see that they do not shorten, actually they just slide past each other using these myosin head and actin binding site. So that is what is given here. Do not shorten but rather pass each other. So the sliding filament theory was given by Huxley, and it states that actin and myosin filaments do not become short but they rather slide past each other. Because of sliding of actin filament over the myosin, the length of I band will change. So in the further questions we'll see how the length change is seen in the filament theory. So this is what happens. There is only sliding. The contraction happens, but there is only sliding of the filaments over each other. So moving on to the next question. The head zone in the skeletal muscle fiber is due to the central gap between myosin filaments in A band, the central gap between actin filaments extending through myosin filaments in A band, extension of myosin filaments in the central portion of A band, the absence of myofibrils in the portion of A band. So let's look at the diagram to explain this. So we have the myosin thick filaments here, and we have the actin filaments here. So actually a sarcomere is present here, okay, that is between the two Z lines. Sarcomere is the unit that is present between the two Z lines, and here we have the myosin thick filament, and we have the actin thin filament here. And so we have been asked about the A band. So let's look at the option: the central gap between myosin filaments and the central gap between actin filaments extending through the myosin filaments in the A band. So the extension of myosin filaments in the central portion of A band, the absence of myofibrils in the central portion of A band. So the apt explanation is that the A band does not contain any actin filament, but it contains only the myosin thick filaments. The solution is going to be the central gap between the actin filaments. So we don't have any actin filament here, and we have only the myosin filament in the A band. So that is what the H zone is all about. H is also called Henson's line, named after the person who discovered this. So the answer is H zone. So the H zone is the central gap between the actin filaments extending through the myosin filaments in the A band. So moving on to the next question. During muscle contraction in humans, the actin filament shorten, the sarcomere does not shorten, the A band remains constant, the H and I bands shorten. So let's look at the contraction procedure. So we have the muscle fibers here. Sorry that the diagram is quite crampy. So let's just see. We have the actin filament, myosin filament, and we have the A band, and we have the H line. Here we have the Z lines. So when contraction happens, the I band is shortened, and also the Z line also comes closer, and that's shortening here. We don't see much contraction, but actually the contraction is there. And also the A band also shortens. I'm sorry, wait a moment. The A band does not shorten. The A band does not shorten because the myosin filament remains constant. Only the actin filament moves about. So the A band remains constant. The I band changes, and the Z line also changes. So sarcomere also shortens because the sarcomere is a unit that is present between the two I bands. So the the Z line. So the sarcomere also contracts. When there is a contraction between the two Z lines, the sarcomere also shortens. So actin filaments shorten, sarcomere actually shortens, so we have to omit this option, and A band remains constant. This is the correct option, and A, H and I bands shorten. Actually the A band does not shorten, so we can leave this option off. So the answer is A band remains same. So we see here A band remains same. Increase in calcium level in the sarcoplasm leads to the binding of calcium to the subunit of troponin on actin filament, and thereby the removing the mask of active sites for the myosin. So the mechanism between contraction is there is a neuron here. So there is neurotransmitter release, and when the neurotransmitter is released, calcium 2+ ions in the sarcoplasmic tubules are released. So what happens? This calcium will go and bind to the troponin, that is C troponin. So this will remove that, and so there is space for the head of the actin, head of the myosin filament to go and bind. So this is where the ATP hydrolysis also occurs. The myosin head now binds to the active site on the actin to form a cross bridge. So there is formation of cross bridge. Since the calcium goes and binds there, there is also the binding between the actin and the myosin. This pulls the attached actin filaments towards the center of A band. The H line attached to these zones are also pulled inwards, thereby causing shortening of the sarcomere. So there is shortening of the sarcomere, i.e. contraction. It is clear from above steps that during shortening of muscle, i.e. contraction, the I band gets reduced, whereas the A band length remains the constant. So moving on to the next question. Which one of the following pairs of chemical substances is correctly categorized, calcitonin and thymosin, thyroid hormones; pepsin, prolactin, two digestive enzymes secreted in the stomach; troponin and myosin, complex proteins in striated muscle; secretin and rhodopsin, polypeptide hormones. So let's discuss them. Calcitonin and thymosin. Calcitonin is thyroid hormone, but thymosin is not a thyroid hormone. Prolactin. Thymosin is basically small proteins found in the thymus, not in the thyroid. Pepsin and prolactin. Pepsin is a digestive hormone, but pepsin is a digestive enzyme. Prolactin is a hormone that causes milk secretion and let down. That is the milk being released from the mother's breast so that the child can feed upon it. The troponin and myosin are complex proteins in striated muscles. Yes, troponin and myosin cause the contraction and they form the important part in the sliding filament theory. Secretin and rhodopsin, polypeptide hormones. Secretin is a polypeptide hormone, but rhodopsin is not a polypeptide hormone. Rhodopsin is present in the eyes and it is a reason for the vision. And so the correct answer is troponin and myosin in complex protein in striated muscles. Troponin is a protein which is found in actin filament, and myosin is a protein found in myosin filament. So both actin and myosin are complex proteins in striated muscles. Thymosin is a hormone secreted by the thymus that stimulates the development of T cells. Prolactin is a hormone released by pituitary gland that stimulates breast development and milk production in women. Rhodopsin, also known as visual purple, is not a hormone. It is a biological pigment in photoreceptor cells of retina that is responsible for the first events in perception of light. So moving on to the next question. Which of the following is the contractile protein in muscle? Myosin, tropomyosin, actin, tubulin. So this is basically a fact. Let's look at the answer. So actin and myosin are not contractile proteins. They are part of the skeletal muscle. We saw in the they are complex proteins involved in the striated muscle. So tubulins are also proteins only. They are also protein, but they are part of the microtubules in the cells. When we see them in the tubulins are mostly found in the cellular level. So when there is cell division, we see microtubules. When examined closely, they are mostly made by tubulins. So myosin is the only contractile protein of the muscle. Answer is myosin. So moving on to the next question. What is sarcomere? Part between two H lines, part between two A lines, part between two I bands, part between two Z lines. We have discussed about this in the previous questions, but just a reminder. So we have the muscle unit here. So sarcomere is defined as the part between two Z lines. Okay, so this is a fact, so we have to remember. Dark bands on the myofibril form A band. So dark band, so there is this overlapping of muscle fibers, so there is a dark band. Middle part of A band contains a light zone called Henson's line, so this is the H zone. There is no actin filament here. Light band on the myofibril is called I band, so we have the I bands here. Middle part of the I band is called Z line. So Z, there is we saw in the other pictures as this, so this is called the Z line. Sarcomere is the part between the two Z lines. So when there is contraction, the sarcomere also shortens, but the A band, the H zone does not change. So this is the part between two Z lines. Which statement is correct for muscle contraction? Length of H decreases, length of A band remains constant, length of I band increases, length of two Z line increases. So we know, as discussed from the previous questions, the A band remains constant, so this is correct. Length of I band increases, no, this is not correct. Length of Z line increases, no, both of them decrease together. So length of H line decreases, this is also correct. So we have two correct options in the given question. So let's look at the solution. We're not sure what it is. So we have the contraction, and so this is the process explained here. Length of the H line is actually increasing, yes. decreasing when calcium ions combine with troponin. Contraction of muscle initiates. So we have the C troponin to which the calcium binds, and there is unmasking. During the contraction, the Z lines come close together and the sarcomeres become shorter. The length of A band remains constant, I band shortens and H band narrows. So the length of H line decreases. Also the length of A band remains constant. So moving on to the next question. Which ion is essential for muscle contraction? Sodium, potassium, calcium, chloride. So we know we have the C troponin to which the calcium binds. So the most necessary thing that is for the muscle contraction is Ca. We should not say sodium or potassium. They are involved in neuromuscular contraction. Chlorine is also involved in various other metabolisms in kidney and then also in neuromuscular contraction. Neuromuscular conduction. So the basic thing that is necessary for calcium, necessary for muscle contraction is calcium. So the answer is calcium. Movement of Ca2 out of the sarcoplasmic reticulum controls the making and breaking of actin and myosin complex. Actomyosin, due to which muscle contraction and relaxation takes place. Albers and Gairogi worked out biochemical events of muscle contraction. So this is not needed. So let's move on to the next question. Characteristic of smooth muscle fibers are. So let's, instead of reading this, let's go to the solution. So we'll let you know what the other features of the muscle are. So we have the cardiac muscle, we have the skeletal muscle, and we have the smooth muscle. Smooth muscles are those which line the body cavities. That is, we are not able to see them, they are inside. They mostly cover the linings of the stomach and all, not the mucosa, the outer part of it. There is smooth muscle. So let's look at the characteristics of each of the muscle. So we, taking a closer look, we see their spindle shape, and they are not branched, and we have single nuclei only. So in the skeletal muscles, we see them as cylindrical, multinucleated, and not branched. In cardiac muscles, we see them as branched, cylindrical, and multinucleated. So smooth muscles are involuntary. We don't know if they are moving inside our stomach or something. So they are basically involuntary. So they have asked about smooth muscle fibers. So the answer is they are spindle shaped, they are not unbranched, they are unstriated, uninucleate, and involuntary. So let's look at the general characteristics of smooth muscle fibers. Cells range from 5 to 10 micrometer, and the diameter is 30 to 200 micrometer in length. Spindle shape, single centrally located nucleus, we have them here. Single muscle tissue occurring with almost every organ, forming sheets, bundles, or sheets around other tissue. Smooth muscle differs from both skeletal and cardiac muscle tissue in structure and function. Sarcomere or myofibrils are not present, therefore not striated. So they are called smooth muscle. So moving on to the next question. The pivot joint between atlas and axis is type of cartilaginous joint, saddle joint, cartilaginous joint, synovial joint, saddle joint, fibrous joint. So the atlas and the axis, they are the first two of the vertebrae. The atlas holds the skull in the place, and the axis comes after the atlas. So we rotate our head towards right and left, up and down. So this is due to the free movement of atlas and axis over the straight axis of the body. So even if you are not able to figure out the answer for this question, so you should use a logical terminology here. The atlas and axis are reason for the movement of the head towards right, left, up and down. So the joint between them should also be movable. So figuring out that way, cartilaginous joints are not very flexible. They the actual movement of head and neck is very complex, and that is not possible much with the cartilaginous joint. Synovial joints are actually very flexible. We have them in the knee, we have them in the tibia and tibia and the fibula. So we know the synovial joints are very good in movement. Saddle joints are present in the tarsals and metatarsals. They are good for movement, but not in the all directions as in head and neck. Fibrous joints are not very flexible, so the answer must be synovial joint. Pivot joint is actually a type of synovial joint. So I just wanted to say, like, most of the people find difficult to answer this question, so I'm giving you a small logic so that you can remember. Pivot joint is a type of synovial joint which provides freely movable between atlas and axis vertebrae of the vertebral column. So this is the atlas and this is the axis. So this has the axis and we have the joint which is synovial joint, pivot joint. So moving on to the next question. Out of X pairs of rib in humans, only Y pairs are true ribs. Select the option that correctly represents the value of X and Y and provide their explanation. X is equal to 12, Y is equal to 5. True ribs are attached dorsally to the vertebral column and sternum on the two ends. X is equal to 24, Y is equal to 7. True ribs are dorsally attached to the vertebrae but are free on the ventral side. X is equal to 24, Y is equal to 12. True ribs are dorsally attached to the vertebral column but are free on the ventral side. True ribs are X is equal to 12, Y is equal to 7. So true ribs are attached dorsally to the vertebral column and ventrally to the sternum. So let's look at the solution. So we have 3 types of ribs here. Basically there are basically 12 types of ribs, 12 pairs of ribs. So we can reject both of these options. So we have 12 and 12. So the first 7 are true. Why do we call them true? Because they are attached to the vertebrae, and anteriorly, in the ventral side, they are attached to the sternum. These are the false ribs. The 8 to 10 is the false rib. There are two pairs of false rib, and also the 11 and 12 are completely floating ribs. They do not directly attach to the sternum. So the answer is there are 12 pairs of ribs, of which 7 is true ribs. True ribs are attached dorsally to the vertebral column and ventrally to the sternum. So the answer is D. So glenoid cavity articulates scapula with the acromion, clavicle with the scapula, humerus with the scapula, clavicle with the acromion. So let's look at the solution. This is a simple fact basically. So this is the acromion process and scapula is the bone here. So scapula, it articulates with the large bone of humerus that is in the upper limb. There is humerus, the head of the humerus goes and articulates in the scapula. So glenoid cavity is the this is like a ball and socket. This is a ball and socket joint, and so the socket is in the glenoid cavity. So what does this do? The glenoid cavity basically attaches the scapula, the bone of scapula, to the upper head of the humerus. So humerus with the scapula is the answer. Glenoid cavity articulates humerus with the scapula. So moving on to the next question. Which of the following joints would allow no movement? Cartilaginous joint, synovial joint, ball and socket joint, fibrous joint. So we discussed in the previous questions through logic. So we know fibrous joints do not allow much of movement. Fibrous joint is attachment between the bones being held together by fibrous connective tissue. For example, sutures. So we know there is skull. So when we open the skull, we have small, it's not a full mass. We have these kind of crack like structures, but the skull does not break when you touch them there. So basically there is fibrous joint when we are born as children. It is weak, and as we grow as adult, it mineralizes. So there is no complete mineralization. So there we can see all the patterns on the skull. So that is called fibrous joint. Which of the following is not a function of skeletal system? Storage of minerals, production of body heat, locomotion, production of erythrocytes. So we know bones are made up of calcium, phosphorus and other organic minerals. So when there is an hormone deficiency, when there is a increased hormone such as thyroid hormone or even vitamin D, so the first affected ones are minerals. So when there is a mineral metabolism impairment, the basic thing that we see is bone, there is a difference in bone structure. So it is actually a storage of mineral. So that's why when we see when there is a fracture or when there is a weakness in the body, the bone also tends to become weak. So the mineral needed for the body is taken up from the bones. That is, it is resorbed and taken into the plasma and blood for other uses. Production of body heat. Let's discuss this later. Locomotion. Yes, if there are no bones, we'll not be able to move at all. If the bones are not attached, if the muscles are not attached to the bones, we'll have no rigid structures. Production of erythrocytes. So we might get confused. Actually, the bones have the bone marrow. The long bones have the bone marrow. We know bone marrow is the major producer of all kinds of cells in the blood. So majorly erythrocyte, RBC. So all these three functions are correct. So what is production of body heat? This is not by bone. This is by adipose tissue and also by liver. So this is not a function of skeletal system. So production of heat is caused by process of metabolism, and it is not by the skeletal system. So thank you for watching the video.